Hi there, thanks for tuning in. I'm Tam Smith with the U.S. Fish and Wildlife Service, Minnesota-Wisconsin Ecological Services Field Office. I work mostly with endangered species recovery, and today we'll be talking about a plan that we recently developed for the Spectaclecase captive rearing and releases. Just a bit of background. The Spectaclecase was listed as federally endangered in 2012, but the host fish was not discovered until 2017. So at that time, since we knew it was possible to rear the species in captivity, we began to plan for rearing the species and for eventual reintroductions. So a group of people from numerous entities worked together to develop a Spectaclecase propagation and reintroduction plan, which is now on the U.S. Fish and Wildlife Service's Spectaclecase website, and the web address is up here. I wanted to thank everybody who contributed to this plan. This plan is intended to explain the rationale for pursuing propagation and reintroduction, how it will be achieved with minimum risks and maximum likelihood for success, how the project will be evaluated, and then, when appropriate, terminated. The International Union of Conservation of Nature, or the IUCN, guidelines were used to facilitate an assessment of ecological, social, and economic risks, and to aid the development of collection, release, and monitoring strategies. These types of planning activities to minimize risks to extant populations and their habitats is imperative. It's imperative to avoid harm to existing populations of target and non-target species and to make efficient uses of our limited resources. The service cooperated with state, federal, and local agencies, universities, and other partners to develop this plan for Spectaclecase, and will cooperate with those partners to implement the plan. The first part of the document lays out the goals. The primary purpose of planned augmentations or reintroductions is to establish free-ranging, self-sustaining wild populations of the species. But in this plan, we dug down a bit deeper and developed some of the specific goals that all contribute to the overarching goal to make positive contributions to Spectaclecase recovery. We came up with 4 main goals. The first goal is to establish baseline data and criterion to help inform reintroductions and augmentations. The second goal is to optimize captive rearing techniques, establish protocols for Spectaclecase propagation and husbandry, and to captively rear the species. Our third goal is to establish self-sustaining populations of Spectaclecase within its current or historical range. And our fourth goal is to resolve key uncertainties to help improve Spectaclecase propagation, augmentation, and reintroductions, and to inform other conservation activities for the species. Each of these goals has step-down objectives and actions, and the plan did not attempt to solve all the objectives and actions, but instead lays them out as targets and things for us to be thinking about as we proceed with activities. After laying out the goals for the plan and some step-down actions, we moved to feasibility and design. The feasibility assessment should cover the full range of relevant biological and non-biological factors. This is where you can identify what's known about things like the basic biology of the species, the habitat needs of different life stages of Spectaclecase, etc., but also make sure that you're considering the host fish needs, genetics, climate change, animal welfare, and also the non-biological side of things, making sure that folks are in compliance with the regulations, that folks have the resources to conduct and maintain these activities, and to think about the social implications of doing reintroductions. Any reintroduction or augmentation has certain risks that it won't achieve its objectives or cause some unintended harm. So the full suite of possible risks, both during a release and after a release of organisms, should be assessed in advance. Risk is the probability of a risk factor occurring combined with the severity of its impact. So here we review the ecological risks, such as will the species have impacts on other species or ecosystem functions, disease risk, such as the possible consequence of disease spread, risk for importing or exporting invasive species and how to minimize this risk, genetic risks, for example, that can result in lower fitness of offspring, social economic risks, For example, will this directly impact people or their livelihoods or affect their public attitudes? Financial risks, where there should be some assurances of funding. There's always a possibility of less or discontinued funding, and the consequences of that should be considered. And the risk of doing nothing should also be considered. Some of these risk factors have very little or poor data, but the lack of that data does not indicate the absence of the risk. Then the plan gets into implementation, which extends beyond the release of organisms. Here we talk in depth about how to select source or broodstock populations, broodstock collection techniques, techniques to rear in captivity, including using host fish and in vitro methods, and various juvenile culture techniques. This section also describes release techniques, disposition of excess progeny, and choosing release sites. In the plan, we provide some criteria and ranking factors to help choose sites for collecting broodstock, for reintroductions, and for augmentations. Here are some of the criteria we developed to help rank and score source or broodstock sites. This is just a brief summary, and more detail is in the plan. Some of the primary criteria included whether or not the site had evidence of recent reproduction, because healthy populations would maintain reproductive capacity. Sites with a positive population growth rate, because they're more likely suitable for supplying Glaucidia than declining populations. The low abundance of invasive species like zebra mussels that could compete for resources because populations could be stressed or unknowingly in decline if overwhelmed with invasive species. We're also looking for genetically diverse populations that are close to the reintroduction sites. Abundance of the spectaclecase, so sites where the species is abundant is more likely suited to supplying broodstock. And ecological similarity to the potential reintroduction sites. A secondary criteria is the ease of access to facilitate efficient collection and monitoring. For selecting reintroduction sites, we considered the species' biotic and abiotic requirements. Some criteria were that the site was within the historical range of the species to reduce regulatory burden and to increase potential for connectivity to other sites, the presence of the host fish, which is necessary for population growth, presence of appropriate substrate, water quality within acceptable standards, distance to currently occupied sites, years since the spectaclecase was last observed live at the site, as those might be more likely to sustain populations than those where the species has not been recently observed. Historical abundance, because those sites might be more likely to sustain larger populations. Presence of other federally listed species to reduce regulatory burden. Proximity to broodstock sites, here we're thinking about genetics and genetic lineages. Abundance of invasive species, specifically zebra mussel densities. Whether or not the site is prone to catastrophic events, including natural disasters and human disturbances. Indicators of mussel community health, and that the cause of decline of spectaclecase at the site, if known, was eliminated or reduced. And last, looking at the percent developed land within a certain distance of the site. We also had secondary criteria for this one too, that made sure that we kept practical needs in our minds as well, like ease of access, site ownership, and ease of monitoring. So this is just a visual from the plan itself to show you the amount of detail that we laid out for each one of these criteria. As released individuals become established, the emphasis will shift to monitoring and to adaptive management based on the monitoring results. Ideally, this is a cyclical process of implementation, monitoring, and adjustments to implementation, and round. This is where we measure the performance against our objectives. In this section of the plan, we discuss data management, population and habitat monitoring at the release sites, including demographic and health monitoring for pilot projects and short and long-term monitoring of the population. We also discuss how to monitor habitat, host fish, and genetics. Hopefully everything goes according to plan, but that's not always the case. So not all releases go according to the plan, and there might be a point when investing more resources is no longer justified despite adjustments to the program. It's important to recognize when to discontinue the programs then. Ideally, one of the first two on here would have happened. The species is recovered and no longer needs further releases or augmentations, or the objectives of the plan have been reached. Other circumstances, though, should be evaluated. For example, the release programs fail over numerous years, and it might be best to reevaluate the program at that time. And so we discussed those different scenarios in this plan. So in today's presentation, I quickly summarized in brief a lot of the things that are included in the plan, but didn't get a chance to get into much detail. So this is just a reminder that the full report is available on the U.S. Fish and Wildlife Services website, and the web address is at the bottom of your screen. Thank you all for watching this presentation. I also want to thank everyone who helped to develop this plan and thank everybody who will be implementing this plan in the future. I'm excited and I'm looking forward to moving Spectaclecase towards recovery. Thanks everyone.